Hello, my name is Ariel Rokem. I'm at the Department of Psychology and at the University of Washington eScience Institute, and I'm here to tell you about work that we're doing on automated quantification and analysis of human-brain connections. The connections between different parts of our brain form networks that are fundamentally important to support complex behaviors that we execute. These networks also change with development and learning, and their properties are important for brain health. And in our work, we look at measurements of these human brain connections with magnetic resonance imaging, or MRI. And the method that we use is called tractometry. This uses the anatomy, the known anatomy of the brain, to generate features, do feature engineering for statistical analysis and for machine learning methods that go from the MRI images to brain connections. And then we take these features and we do a variety of, we use a variety of machine learning methods in order to do predictions and inference based on those about what's important about these features for a variety of different behaviors and other properties of different individuals. So let's start with the MRI, magnetic resonance imaging method. Here on the left, you see an MRI, and this person lying on this bed is about to enter the MRI. And when they go in there, their brain can be scanned, and we can tune the magnet in a variety of different ways in order to generate different kinds of images. The images that we look at are called diffusion-weighted images or diffusion-weighted MRI. Diffusion-weighted MRI, a magnetic field gradient is applied across the sample in a variety of different directions. And then this makes the measurement sensitive in each location in the brain, sensitive to the direction of the diffusion of water in that location. And this lets us measure the properties of brain connections, and that's because the fascicles that form the brain connections, made out of millions and millions of nerve fibers, those fascicles restrict the diffusion of water so that the water diffuses more readily along the length of the connections than across the boundaries of the brain connections, the nerve fibers. And so taking this diffusion-weighted measurement, we can model the process of diffusion in every voxel. So here we're looking at the measurements in multiple different diffusion directions in one voxel in the brain. And the distance from the center of the point cloud is an assessment of the amount of diffusion in that direction. And we can simplify these measurements using a model. Here we're using a diffusion tensor model represented by this 3x3 two-way tensor. And here is a graphical representation of this tensor, which gives us a smoother representation of the original data as kind of ellipsoid. Based on this model, we can assess different physical properties of the brain in different places. For example, mean diffusivity gives us an assessment more or less of the density of the tissue in different places. You can see that in parts of the brain where there are ventricles, there's much more diffusion, and that's because the water molecules are not restricted in their diffusivity. On the other hand, inside the core of the white matter, there's a lot of tissue, and that restricts the motion of water. Another kind of physical property of the brain or assessment based on this model is how much diffusion is there in some directions relative to others, how anisotropic is diffusion. And you can see that inside of the white matter connections, for example, here in the corpus callosum that connects the two hemispheres, the diffusion is very oriented, it's very directional along the length of the fibers. So this gives us an assessment of the directionality of diffusion. And then finally, there's the principal diffusion direction. This is the direction in which this ellipsoid is pointing. Where is there most diffusion in what direction? Again, inside the corpus callosum. And so we encode this in this image as an RGB based on the vector of the principal diffusion direction. For example, you can see that inside of the corpus callosum, there's a lot of diffusion right to left, red. Along other tracks, there's a lot of diffusion front to back, encoded in green. And then here in this part of the image that's blue, there's a lot of nerve fibers going up and down, encoded in blue. So in tractometry, we use information about the anatomy to engineer features. And this is a multi-step process. In the first step, we generate estimates of brain connections. Then within these connections, we find the major white matter pathways. We assess their physical properties, and then we can use that to compare across individuals, between different groups, individuals, and so on. So let's go through this step by step. In the first step, we use computational tractography to generate estimates of brain connections. This is done by looking at the properties of the diffusion in individual voxels and connecting between them. Here we're focusing on a particular part of the brain. And in this part of the brain, we fit a tensor model in every one of the voxels, and then we can follow the direction of the tensors in each voxel in order to propagate streamlines like this one that form estimates of the connections. If we put the seed for this tractography process in a different voxel, then it will generate a different estimate in a different part of the brain. And we can repeat this process many, many times by placing seeds all over the brain's white matter, and we get a lot of these streamlines. We can then say we'd like to select only streamlines that pass through some particular part of the brain. For example, here in yellow is the corpus callosum. And we've asked here the algorithm to give us only the streamlines that pass through the corpus callosum in the front of the brain, giving an estimate of a particular anatomical connection. And we use that in order to find major white matter pathways that are known from the anatomy within the tractography results. We do this by placing waypoint ROIs and asking for streamlines that only pass through these waypoint ROIs. And we've developed software. PyFQ, John Kruper, whose picture is up here, is the main developer of this software. And here you can see how this software finds the corticospinal tract. It goes up, down, and connects the cortex to the spine. If we place the waypoint ROIs in other places in the brain, we get this arcuate fasciculus. It's another bundle of fibers that connects the temporal cortex to the frontal cortex. And then another example, another set of waypoint ROIs gives us the inferior frontal occipital fasciculus that connects the occipital cortex, the back of the brain, to the front of the brain. And we can do that for many different known anatomical connections. Once we do this, within each bundle, we can assess the physical properties of diffusion within that bundle. And I wish that I had come up with this acronym, but Derek Jones and colleagues came up with this in 2005. This is called Pointwise Assessment of Streamline Tractography Attributes, or PASTA. So here's how PASTA works. Let's look at the corticospinal tract as an example. We divide up the corticospinal tract into a collection of nodes, and then we extract, for example, here on the right, the fractional anisotropy properties along each one of these nodes. We do that for the first node and place that, and then we do that for the second node, and we take that one, and the third node, and so on. And if we do this for, well, let's say that we divide up the corticospinal tract into 100 different nodes, and we do that for every node, we get these track profiles, what we see here on the right. And we can do that for every different anatomical bundle in the brain that we find using PyFQ. Finally, we can use this information to compare across individuals, between different groups, between individuals and groups, and so on. One approach that we can take to that is the group comparison approach. Here, for example, is the track profile from a group of patients with amyotrophic lateral sclerosis, or ALS, depicted here in blue, and a group of matched controls in red. And this is the fractional anisotropy measured along the corticospinal tract. ALS is a disease that attacks primary motor neurons. And so it has an effect on the physical properties of the corticospinal tract. And we can compare these groups to each other and then do an assessment of how statistically significant the difference is in different locations, and find that there are some parts of the corticospinal tract where there are significant differences between these groups. Another approach to this is to take a prediction approach. In this approach, we lay out properties of different individuals. For example, here, let's say Y1 represents the age of the first individual, Y2 may represent the age of the second individual, and we try to fit a linear model to the diffusion data. We solve for beta here to find the best combination of track properties that best predicts the age of every individual. And so each row in this big X matrix here, each row represents a single individual, and each column represents the physical properties, for example, FA in a particular node within a particular bundle. So for example, here we're looking at the inferior frontal occipital fasciculus along its length, and this might be the FA in the very end of it, and then we march along here, putting in the FA along in different parts of the bundle. And we do this here for a different bundle, maybe some part of the corpus callosum, and here maybe the arcuate fasciculus. You can see. So all of the diffusion properties in all of the different bundles go into, for a single individual, go into each row of this matrix. And then we can fit a model that fits the entirety of the data so that we find weights that overall give the best prediction of age of the different individuals. Now, Adam Ritchie-Halford, who is a postdoc in my group, developed an approach to this. So here again is this model. We're trying to estimate Y, say biological age, from the diffusion properties. Here's next by solving for beta. So beta hat is an estimate of the weights as our best guess for the weights, such that this linear equation provides the least error. And the problem that we have is that usually we have a lot more features. This matrix is very, very wide than we have individuals. We can make these measurements in many, many different bundles. So we get thousands of these features, and we'll have dozens, maybe hundreds in some cases of individuals. And so what we do is we apply regularization. So this equation here describes regularization penalty. This is called the lasso regularization penalty. Here is the least squares penalty in the middle. So we want betas that fit the data well, so as to generate small or least square errors, but also at the same time, this penalty here, parameterized by lambda, is the sum of the betas. So we want a solution that both fits the data, but also gives us betas that are the sum of which is as small as possible. You can do something a little bit more complicated. This is called the elastic net. This combines the lasso regularization over here on the right, and then also the ridge regression penalty, which is the same, but instead of looking at the sum of the betas, you look at the sum of the square of the betas. So this is called elastic net. It combines between the lasso and ridge regression, and the degree to which you're either closer to lasso or ridge regression is parameterized by this alpha. So how much is the relative weight of the ridge penalty versus the elastic penalty. Now, Adam took a method that Noah Simon, who's a colleague of ours at the University of Washington, Department of Biostatistics, developed. That's called sparse group lasso. Sparse group lasso, and here's the penalty for the sparse group lasso. What it does is it takes into account the fact that the different features come from different groups. So you can see here, this looks a lot like the ridge penalty up here, and this looks a lot like the lasso penalty. This, in fact, is the least squares penalty, but we've divided up the matrix up based on the columns that belong to these different bundles. So this is a group, and this is a group, and this is a group. And we apply a group-wise penalty in the ridge regression. And this group-wise penalty, what it tends to do is it tends to select entire groups for inclusion in the model or for exclusion from the model. So this has the property of taking into account the anatomical structure. And it turns out that helps fitting these models. So here's an example from data from the Cambridge Center for Aging and Neuroscience. They measured diffusion MRI in a group of almost 700 individuals. And here what Adam has done is fit this model using cross-validation, fit this model to the diffusion properties from the brains of these individuals. So here on the x-axis is their actual age, and here on the y-axis is their predicted age. And you can see that the model fits very, very accurately, and this is assessed in cross-validation. In addition, this gives a relatively interpretable result in the sense that the weights that we get out of this model can be projected back onto the anatomical structures from which this data came, so that we can interpret and say which of these white matter tracks or bundles plays an important role in predicting age, and by that assess which of these changes, which of these tracks and which of their physical properties change dramatically with age in order to allow this prediction. So to summarize, we in our work, we take MRI images, we extract features from these images, And we use machine learning models, in particular, the sparse group lasso that I just described, to fit the data. I'd like to thank my colleagues who work on this, in particular, highlight John Kruper, who's developed PyFQ, Python automated fiber quantification, and Adam Richard Halford, who's done the work on sparse group lasso that I just described. I'd like to also thank our sources of funding and support for this work, and I'd like to thank you for listening.